Welcome, everyone. We have Dr. Liberman with us today. He earned his bachelor's in biology at Harvard and followed by his Ph.D. in physiology at Harvard as well. And he's currently a professor in otolaryngology and head and neck surgery at the Harvard Medical School and a senior scientist at the Eaton-Peabody Laboratories. And he has a wonderful talk for us today. Thank you for joining
Just
us.
happened. Thanks for inviting me. The title of my talk today is Hidden Hearing Loss, Facts and Fantasies. So when most of us think about age-related hearing loss, ARHL, or noise-induced hearing loss, NIHL, we think about audiograms like this. This is a conventional way of measuring hearing loss. We have here some mean audiograms from publication we just put together with our group. These are some normal aging audiograms as a function of age here, color-coded, and some data we got from the Million Veterans Program showing the same range of ages. And as no surprise to any of you, the standard sloping high tone hearing loss of normal aging, which is accelerated, especially in the 4 and 8 kilohertz region in this veterans population, almost certainly reflecting noise-induced hearing loss. You can see that a little more dramatically if we just compare right on top of each other this normal aging group and the veterans group, 60 to 64 years old. You can see the appearance of essentially the 4 kilohertz notch, although there's also a lot of loss at 8 kilohertz. So the first chapter of my talk, I'm going to review what I think I learned years and years ago about the functionally important structural changes underlying this kind of permanent threshold shift, that is conventional hearing loss as it's measured in the audiogram. So many, many years ago, for my thesis, almost 50 years ago, I was in a lab with Nelson Kiang, who was a pioneer in recording from single auditory nerve fibers. And my thesis was to record from single auditory nerve fibers in the anesthetized cat, recording from the central axons. As you all know, each auditory nerve fiber has a cell body in the spiral ganglia, a central axon that goes to the cochlear nucleus, and a peripheral axon that innervates a pretty much single hair cell in the organ of Corti. So these sharp electrode recordings from the central axons of auditory nerve fibers gave us really an unparalleled granular view of the functional state of the organ of Corti. When you're recording from one of these central axons, you can put in tones of different frequencies and see how intense you need to make the tones to cause the neuron to discharge. And one of the classic response properties is the tuning curve of a single auditory nerve fiber, showing you one high frequency fiber here and one mid to low frequency fiber here, superimposed on a graph of the tips of the tuning curves of maybe 200 fibers we recorded from a single animal. So higher sound pressure levels are up here and lower sound pressure levels are down here. The best frequency or characteristic frequency that we record down near 0 dB SPL obviously tells us where along the cochlear spiral this fiber is contacting an inner hair cell, and the minimum envelope of this cloud of points has to look a lot like the audiogram. So my thesis involved looking at acoustically traumatized cats. We anesthetized them, overexposed them to narrow bands of noise for a couple of hours at levels between about 100 and 120 dB. Waited a few weeks so that the temporary reversible aspects of that threshold shift would disappear. Record from the auditory nerve, and then sacrifice them and do classic pseloidin sections with H&E staining. So over here, we see one animal. This is where the normal thresholds of these auditory nerve fibers ought to be, and each point is a different auditory nerve fiber. So you can see this exposure has caused a huge permanent threshold shift, and not surprisingly, there's a lot of hair cell loss. So this is a classic cytocochleogram. The black regions are where the cells remain, 3 rows of outer hair cells, 1 row of inner hair cells. And the gaps here are where the cells are missing. In this picture over here, where you can see the outer hair cells are missing, we're probably in this region here. So not surprisingly, in regions of outer hair cell loss, there's increasing threshold shift. But what was dramatic over and over again in these animals that we recorded from is that many, many times we could see significant permanent threshold shift, as shown here, that a few weeks after exposure to this narrowband noise here, there's obvious threshold shift in the mid-frequency region, more or less appropriate to the spectrum of the noise that we put in. But there's really no significant hair cell loss outside of the very base. This is definitely noise-induced. We call it the tonotopically inappropriate lesion. It wasn't present in the other ear. And there's a whole interesting story about what this probably represents, but we're not going to talk about that today. So around the same time, people were beginning to use the scanning electron microscope to look at animals with permanent noise-induced threshold shifts, and there were lots of studies, including one that we did, that showed there was tremendous damage to surviving hair cells. Here we see an inner hair cell with fused stereocilia, one with missing stereocilia, and one here where all of the tall row of stereocilia on an inner hair cell are either missing or fused into this bundle. For a variety of reasons, we chose to assess this stereocilia condition in the light microscope. We found that in cats, if you stained them with osmium and did surface preparations, you could resolve the individual stereocilia. You could actually count them. It's pretty easy to see even in this picture, and even easier to see when you're focusing up and down on the microscope, the extent to which there's tremendous disarray and loss of stereocilia in the exposed ears, and nice regular pattern of a couple of rows of hairs on each inner hair cell in the control ear. So in a very large population of these cats, we had a completely unbiased observer literally counting on every single inner hair cell the number of stereocilia that were still present and normal, and plot that as a function of frequency. So remember, in this ear, there's absolutely no hair cell loss. This is the pattern of threshold shift. And you can see there's a remarkably good correspondence between the region of the organ of Corti where there's terrible stereocilia damage, or in this case, moderate stereocilia damage on inners and outers. And it seems in some sense that it ought to be able to explain this threshold shift. I'm just going to show you one other example where there was a much larger permanent noise-induced threshold shift, and correspondingly, there's a much larger region of the cochlea where there's much more damage. Virtually all the tall stereocilia on the inner hair cells were damaged, and there was also terrible disarray and loss of the stereocilia on the outer hair cells. So suffice it to say that after a large number of these cases, we convinced ourselves that once you had looked at the pattern of hair cell loss and the pattern of stereocilia damage, you could pretty well predict the pattern of permanent threshold shift in auditory nerve fibers. So we're quite confident that we understand the functionally important changes in noise-induced permanent threshold shift. So a number of years later, my colleague Sharon Kujawa and I did a study. It was really her idea. She was very interested in the interaction between noise and aging, wondering if you overexposed an individual subject to noise at a young age and then let them age, how would the cochlea fare compared to an unexposed littermate? These experiments were done with mice. What we were really expecting to see was that there would be additional hair cell loss as the animals aged. But as you can see here, when you look at an animal that was just age only versus an animal that was exposed to a noise that caused a small permanent threshold shift at a young age, but no hair cell loss, there's no or minimal increase in hair cell loss, but you can see a dramatic fallout of spiral ganglion neurons. It looks very dark here, but at high power, you can actually count them. And you can see when you look at these histograms that although there's a little bit of a additional loss of outer hair cells when animals were allowed to age after an early noise exposure, the really dramatic difference was the loss of spiral ganglion cells. So this is a kind of primary neuronal degeneration. The hair cells are surviving, but as they age after this noise exposure at a young age, um, they begin to lose their afferent innervation as spiral ganglion cells drop out. So although this was surprising, it really caused the light bulb to go off in my head because of experiments we had done years before on temporary threshold shift. So that brings us into chapter 2 of my talk, where we talk about hidden hearing loss, temporary threshold shifts, and cochlear synaptopathy. So back in the day, when I was recording from the auditory nerve of cats, I was interested in permanent threshold shifts. So in addition to studying permanent threshold shift, we looked at animals. We were interested in the functionally important structural changes underlying temporary threshold shifts. So we took normal animals and we recorded from the auditory nerve for a while, and then we acutely exposed them to high-level sounds loud enough to make, again, a 30 or 40 dB threshold shift, but now it's reversible. So if we had waited a week or two, this would have completely disappeared. So here's one animal, here's another animal, here's a third animal, and when we did the histology immediately after these experiments, you can perhaps see that there's absolutely no damage to the stereocilia. So it's a fundamentally different um, ball of wax as to what's causing the threshold shifts in temporary threshold shift versus permanent threshold shift. So Don Robertson was studying the same phenomenon. Here's one of his pictures showing the swelling of afferent terminals underneath the inner hair cells. We could see this in the light microscope. You could see it in the electron microscope. In the 80s, people assumed that this kind of swelling must disappear and there must be no residual damage because the thresholds return to normal. But as we'll see in a minute, I knew that you could lose a lot of auditory nerve fibers and have no threshold shift. And so I stored away the idea that perhaps not all of these swollen fibers actually did recover. And we then pursued that because in the early 2000s, a group had shown that there was a way to immunostain synapses in the inner hair cell with a protein that's in the presynaptic ribbon called CTBP2 and an antibody to glutamate receptors. You can visualize in the confocal microscope these synapses. Since every auditory nerve fiber is unbranched and contacts a single inner hair cell by a single synaptic complex, you can count the number of surviving auditory nerve synapses very rapidly in the confocal. And here are some pictures of a normal mouse showing 1, 2, 3, 4, 5, 6 inner hair cells stained with myosin 7A. And these closely opposed red and green dots, each of which is a synapse. And it's very easy to count the synapses, and it's very easy to see that if you overexpose an animal, as we did here, to produce an acute ABR threshold shift of about 40 dB or 30 dB by distortion products, which is a reversible lesion, such that 8 weeks later thresholds would have returned to normal. If you look at these ears immediately, 24 hours or 1 week after exposure, you can see there's a huge loss of synapses. So throughout the whole basal half of the cochlea, there were about half as many synapses on inner hair cells as there were in a normal ear, shown by these black symbols here. So in this plot, we're going out to 1 week post-exposure, but since this early study, we've gone out to 1 month post-exposure and 6 months post-exposure and 1 year post-exposure, and the damage is permanent. And if you look at, instead of looking at thresholds, so there's no threshold shift. By a few days post-exposure, the thresholds return to normal. But if you look at superthreshold amplitudes of either distortion products or auditory brainstem responses, you'll see that although by 8 weeks post-exposure, the superthreshold amplitudes of distortion products recover, the superthreshold amplitudes of ABR do not recover in the high frequency region of the mouse cochlea. So this permanent reduction in ABR amplitude is consistent with the idea that you've lost a tremendous number of synaptic connections. And in fact, in this paper led by Sharon Kajawa, we showed that you could predict the number of synapses per inner hair cell from this wave 1 superthreshold amplitude, so long as you were looking at ears that had no threshold shift. Of course, if you have threshold shift, that's another reason why ABR amplitudes would be reduced, and those ears give you a faulty impression of how much synapse loss there is just by looking at wave 1 amplitude. So this is an issue that I think has generated a lot of confusion, that people think that wave 1 amplitude itself, with no other caveats, is a marker of synaptopathy, but it's only a marker of synaptopathy if you have no permanent threshold shift. Most convincingly, if you wait long enough, here we have post-exposure time on a log scale, and we see that the synapses are lost immediately post-exposure and don't really change very much with time. If you count the ganglion cells, you have to go out to 2 years post-exposure, at which point in time the loss of ganglion cells is about 50%, which matches the original loss of synapses. So the idea is the synapses are lost immediately, but it takes a very long time for the ganglion cells and central axons to degenerate. Now, we've said that this loss of synapses doesn't raise thresholds. In fact, we've known that for a very long time. These are the results of an experiment done 70 years ago by Hal Schucknecht here at the Mass Eye and Ear, who took cats and made surgical lesions of the auditory nerve, creating at least 50% loss of spiral ganglion cells throughout the whole cochlea, leaving the hair cells normal. And these were behaviorally trained cats, and he showed that thresholds were completely unaffected. However, our working hypothesis has always been that this loss of auditory nerve fibers must compromise intelligibility of complex sounds like speech, especially in noisy environments. So now I'd like to talk a little bit about some experiments that we and others have done trying to rescue this synaptopathy. And the key, one of the key molecules in this story is the neurotrophin NT3. So here I show pictures from a reporter mouse, where shown in this blue color from an alkaline phosphatase reaction from a reporter mouse is the expression levels of NT3 in the cochlea and the vestibular organs as a function of age from postnatal day 5 to postnatal day 130. You can see in the neonatal ear, this NT3 is widely expressed throughout the organ of Corti, and it becomes concentrated in the inner hair cell area, both in the inner hair cells and supporting cells, as the animals get older. And there also becomes an apex-to-base gradient. As you can see, there's a lot less neurotrophin released in the base of the cochlea, which is where this synaptopathy is most prevalent. So for a variety of reasons, we thought it was likely that NT3 might help these synapses recover or regenerate after this exposure. And so we did a series of experiments where we took mice, measured their hearing traumatized them with this synaptopathic exposure, and then 24 hours later, opened up the middle ear space and put NT3 in a slow release gel on the round window, and then waited a little bit more than a week to do functional measuring again and count synapses. And indeed, in these experiments, when we used vehicle only down here or a low concentration of NT3, we didn't see any synaptic recovery, but when we used the high concentration of NT3, we could effect almost complete reversal of this synaptopathy. But this is what we saw one day post-exposure. We tried a number of other experiments where we extended the trauma treatment interval to 1 week, 2 weeks, and 4 weeks post-exposure. And I'm showing you here the 1 week and 4 week data. And as you can see, we cannot get any recovery when we introduce this NT3 onto the round window 1 week post-exposure, even though we were able to do it multiple times when we did the intervention at 1 day post-exposure. So our hypothesis in this was that what is perhaps going on is that 1 day post-exposure, most of these synaptic terminals are still present, but by 1 week post-exposure, they've perhaps died back to the habencular region where the fibers become myelinated, and that is a barrier too high for the neurotrophin to have its ameliorative effects on this synaptic recovery. So that was the hypothesis, but you can't really solve that. You can't really see these unmyelinated terminals very well at the light microscopic level. So we decided to move to ultrastructural studies to try to figure out what's happening between 1 day post-exposure and 1 week post-exposure in this synaptopathy model. So we used a technique called FIB-SEM, focused ion beam scanning electron microscopy, which you'll see what the power of that is in a minute. So we took mice and we exposed them to this 8 to 16 kilohertz noise band for 2 hours that causes our classic synaptopathic effect on the basal half of the cochlea. We reacted the cochleas with heavy metals. We map them in the way we would normally map them. We can cut out these pieces, which are actually embedded in plastic, put them on an ultramicrotome, cut 1 micron sections to get at the right place, and then take them to the FIB-SEM machine where we're going to essentially mill our way through the inner hair cell area, imaging it, looking at it this way, and create serial sections through this key region underneath the inner hair cell where all these unmyelinated terminals are. So I'm going to show you a movie of what it looks like when we follow one of these fibers in serial section. So just to orient you, so at this point we are down in here near the basilar membrane. This is actually a bit of the pillar foot. This is the nucleus of a supporting cell. And this is a bundle of nerve fibers coming through the habenula. But we're going to follow this particular nerve fiber here through serial sections. You can see the mitochondria here, the pillar cells. Here's the nucleus of another supporting cell coming in. That's an efferent terminal. There's another supporting cell coming in. Here's an efferent synapse on this fiber, another efferent synapse on this fiber coming up toward the inner hair cell. Still not to the inner hair cell yet. Lots of here's the inner hair cell, and the synapse is right there. So that's what a normal fiber looks like. Now, from a synaptopathic ear, 1 week post-exposure, at a time where we thought perhaps this entire unmyelinated segment had degenerated. So here we are. The supporting cells look normal. This fiber looks completely normal. The mitochondria look completely healthy. There's another supporting cell coming in. There are efferent fibers, just as in a normal ear. There's going to be an efferent synapse on this in a moment. Here's the efferent synapse on that fiber. Here's the inner hair cell. And look right here, it touches the inner hair cell, but there is no synapse. So there's no presynaptic ribbon and there's no postsynaptic specialization. So to our great surprise, what we discovered was that the unmyelinated terminals are all still there. Most of them are still touching the inner hair cell. So this is the control ear. All these green fibers are making synaptic contact. These red fibers, even in a normal ear, a couple of fibers don't make synaptic contact. But 1 week post-exposure in this synaptopathic model, we still have fibers that contact the hair cell, which fits with the 50% loss, but 50% of the fibers don't make synaptic contact. They touch the hair cell, but they don't make synapses. Very surprising. And very surprising that even given that fact that the dendrites have not died back, that the neurotrophin treatment, which worked at 24 hours post-exposure, doesn't work at 1 week post-exposure, even though it looks like all the unmyelinated dendrites are still there, and about half of the non-synaptic ones are actually even still touching the hair cell. Okay, so the last chapter brings us to the human story. So I've been fortunate to be at the Mass Eye and Ear, where for decades, long before my time, people were collecting human temporal bones and analyzing them in a temporal bone otopathology lab run initially by Hal Shuknecht and then Joan Adol and Samuel Merchant. And so all of the infrastructure for gathering human temporal bones was present. Before I get into our results there with the human temporal bones, I want to just point out that, as you probably know, this idea of synaptopathy and hidden hearing loss have really gone mainstream in the sense that there was recently a huge lawsuit against the 3M company who made these earplugs that were supposed to work for soldiers in active combat. And a lot of people complained that they had trouble hearing in noisy environments. And they would go to otolaryngologists, otologists, audiologists, have an audiogram taken. The audiogram would show that there was no threshold shift, there was no conventional hearing loss, but they were actually diagnosed with something called hidden hearing loss, which was the idea that even though their thresholds hadn't shifted, they perhaps had synaptopathy, and that was the reason why they had their complaint of not being able to hear in a noisy environment. So it's obviously an important question to figure out to what extent are all these phenomena that we've seen in mice and guinea pigs. I didn't show you our guinea pig data, and other people have seen in chinchillas and other animal models. To what extent is all this present in human? So we decided to take newly acquired human temporal bones, and instead of processing them as psilocybin embedded blocks and cutting serial sections and staining with hematoxylin and eosin, we thought we would just microdissect them as we do animal bones and see if we could get the same kinds of immunostaining working. Because if you want to count hair cells and neuronal elements, you can do it a lot more accurately and a lot more comprehensively along the cochlear spiral if you're able to do it in these whole mount preparations. So here you can see we've micro dissected out all the pieces of a human cochlea. We've superimposed on it the Greenwood map, so we nominally know the best frequency region in kilohertz of each region, and we can look anywhere we want. Here I show you just a neurofilament stain. You can easily see the region under the hair cells here and the region of the osseospiral lamina where the peripheral axons of auditory nerve fibers are running. Here we show you in a little bit higher power view the region under the inner hair cells, the region under the outer hair cells, the tunnel, and the radially directed peripheral axons of auditory nerve fibers. We can use 4 different immunostaining channels. So in addition to neurofilament, here I show you we're adding cell mask, which is a lipophilic fluorophore, which does a fabulous job of staining myelin. And of course, we can use myosin 7A to label hair cells. I'm going to show you some hair cell data first. So we have collected now 30 or so of these human temporal bones from the morgue at the nearby Mass General Hospital. And we find that if the postmortem time is less than 12 hours, we can quite routinely get very nice staining with these 3 markers to allow us to count peripheral axons, inner hair cells, and outer hair cells. So here are 3 cases. I'm showing you the patterns of loss of inner and outer hair cells. So here is what happens with aging. So we don't have audiometric data on very many of these cases, but we have their medical records. And their medical records tell us that they never sought the care of an audiologist or an otologist. So based on that scanty evidence, we call them normal aging. You can see, obviously, there's a tremendous loss of hair cells, not surprisingly, more loss of outer hair cells than inner hair cells. In addition to that, we can get ESPN to work as a stereocilia marker. It's an actin crosslinker, and we have, I think, pretty convincing evidence that in addition to the loss of hair cells, there's tremendous stereocilia damage on the hair cells of older people, which almost certainly must also contribute to the conventional threshold shift. But in addition to counting hair cells and rating stereocilia in these human temporal bones, this is just to remind me to point out that we believe it's the hair cell loss and the stereocilia damage that accounts for most of this pattern of high tone hearing loss, as well as the extra noise-induced hearing loss in the aging veterans. But in addition to counting hair cells, we can zero in on the osseospiral lamina. We can take a focal series through the osseospiral lamina, and then using various conventional software packages, we can virtually reslice those Z-stacks through the osseospiral lamina, and we can count myelinated fibers. We tried using CTBP2 and counting synapses, and we can make it work, but I trust these counts of peripheral axons much more because I think these myelinated segments of the fiber are a lot less susceptible to postmortem artifact than changes in the inner hair cell cytoplasm. So the data that I'm going to show you are based on counting peripheral axons, and it's not unlikely that there's even more changes in synapses because there's probably a delay between the synaptic loss and the peripheral axon loss. So here's the summary plot from 28 of these humans that we call normal aging because they never sought out the care of an audiologist or an otologist. So each point is from a different individual. They're obviously plotted as a function of age, and we're showing inner hair cell survival, spiral ganglion cell survival, and by auditory nerve fiber, I mean for the peripheral axons of auditory nerve fibers. We are measuring at all the audiometric frequency points from 250 hertz to 8 kilohertz, and we're averaging the loss across all those audiometric points. So in this particular case here, you can see that averaged over all the audiometric frequency points, there was only 5% loss of inner hair cells, but there was 70% loss of peripheral axons. You can also see that there were no exceptions to this loss of peripheral axons with aging. So if this population of 28 ears from the Mass General morgue is representative, it means that all of us, by the time we reach age 60, are going to have lost at least 40 to 50% of the peripheral axons that used to innervate inner hair cells. Most of the ganglion cells survive. So what's happening here is they're losing their peripheral axons. The loss of the cell body in the central axon is very slow. But these are not normal hearing. These are normal aging, which means there's a lot of hair cell loss. I already showed you that. And what this shows you is in a much larger population, there's a very high correlation between the amount of peripheral axon loss and the amount of outer hair cell loss. Much less correlation to the loss of inner hair cells, because as I said, we maintain our inner hair cell population. We're just losing a lot of the innervation of that surviving inner hair cell population. So losing outer hair cells is going to raise thresholds because the outer hair cells are the cochlear amplifiers. So what this means is that as thresholds go up in a typical normal aging human, if this is all representative, that there's sort of a proportional deafferentation of the surviving inner hair cells. And we think that our hypothesis is that both the outer hair cell loss and the auditory nerve fiber loss contribute to problems with word discrimination. And so the last point that I want to make in my talk is in a large population of cases that we've now analyzed with all these different etiologies, we have 135 cases that we have completely analyzed the histopathology, and we also have word scores. Not surprisingly, there's a very strong correlation between the PTA, the mean thresholds, and the word scores. That's been known for a long time. We see that reflected in these data. But what we're interested in is to what extent does how do we explain the variation in word scores among people with the same PTA? And our hypothesis going into it was that the most important histological predictor, once thresholds have been taken into account, is the peripheral axon survival. And indeed, when we do a multivariable linear regression on this data set, and ask if we use all the histopathological measures, including strial survival, which I haven't shown you, peripheral axon survival, spiral ganglion cell survival, inner hair cell survival, outer hair cell survival, and we try to predict the word score. Once we put in the pure tone average, the only other histopathological predictor that improves the prediction of word score is the survival of the auditory nerve peripheral axons. So, last question. It's pretty clear we believe from our data that in normal aging humans with the typical pattern of high tone hearing loss, that is accompanied by a lot of deafferentation of surviving inner hair cells. But is there synaptopathy in humans with normal audiograms? So in a very large study that's really been led by my colleague Stéphane Maison, We have looked at a lot of people with normal audiograms. We put insert earphones in with electrodes, tiptrodes, so that we're recording the ECOG essentially from the ear canal, get a pretty nice big wave one. And given the animal work, which suggested that if thresholds were normal, wave one could help us diagnose cochlear synaptopathy. We've measured that, and then what we've done in addition is give them a very difficult word test. So this is not words in quiet. This is words in very difficult listening conditions. And here are the results to date of a very large study where we're looking at the correlation between wave one amplitude and word score. And our hypothesis was that bigger N1, bigger wave one means more synaptic survival, which ought to be correlated with better performance on this very, very difficult word discrimination test. And indeed, there is a highly significant correlation, but you can see that there's still a lot of spread in the data. So our answer to the question of is there synaptopathy in humans with normal audiograms, we think the answer is yes, almost certainly, but we can't diagnose it yet because obviously wave one amplitude alone is too highly variable to come to a decision on a person-by-person basis. So in summary, noise-induced permanent threshold shift is due to hair cell loss and stereocilia damage, whereas noise-induced temporary threshold shift is not. TTS-producing noise can cause permanent synaptopathy followed by slow spiral ganglion cell death. Synaptopathy can be rescued with NT3, but only at short trauma treatment intervals. Auditory nerve fiber terminals survive for months after noise-induced synaptopathy. Human ears show age-related synaptopathy that contributes to poor word discrimination. And there is synaptopathy in humans with normal audiograms, but at present it's impossible to diagnose. So I just end by acknowledging the many, many members of the Eaton-Peabody lab. Here's a picture of us all in 1984, and I didn't make it into this picture for some reason, but it's grown a lot, and many, many members of the Eaton-Peabody lab contributed to all this work. So with that, I will thank you for your attention.